our second presenter for today, who is Cecilia Roldan-Kozis. She's a rising high school senior at Newton North High School. Thank you.
All right, good afternoon. Good afternoon, everybody. As I've been so kindly introduced, my name is Cecilia Roldan-Kozis, and I'm a rising high school senior at Newton North High School. And today I have the opportunity to share with you all about the research I did in the Concelis lab this summer, which is investigating scribble activity in T-cell differentiation of hematopoietic stem cells. So first, I want us all to get some base information on what a hematopoietic stem cell is. I think we've all heard of stem cells. They proliferate in the body. They're able to create cells from multiple lineages. And hematopoietic stem cells are the cells which create all the lineages for your blood. So that includes your red blood cells, your white blood cells, and your platelets. So these hematopoietic stem cells are found in your bone marrow, and they proliferate based on signals they receive from your body. In many different diseases, transplanting these hematopoietic stem cells, or HSC as they're also known, is curative, since you're replacing damaged or ineffective hematopoietic stem cells for healthy ones. However, if immune reconstitution is impaired, then this can often lead to worse patient outcomes. Impaired immune reconstitution is referring to when your immune system is wiped out, like from a bone marrow transplant, for example. Our lab wanted to see if we could stop this from happening, which brings us to the scribble scaffolding protein. So scaffolding proteins interact with different signaling pathways to form complexes. And scribble in particular has many roles, but it aids in helping these hematopoietic stem cells out of their senescent gap 0 phase, which refers to a cell's resting phase, where it does not proliferate and preserves its activity. So scribble, alongside being tumor suppressor, is helping these hematopoietic stem cells, which aids hematopoiesis, the production of blood cells. We wanted to know a little bit more about scribble. In the past, our lab has found that when we inflame scribble-deficient mice with poly-IC, a chemical, often these scribble-deficient mice will have improved hematopoietic stem cell fitness. So as you can see here, we took some scribble knockout in wild-type mice. We used poly-IC to inflame them. We dosed them 6 times, and we analyzed the leukocyte count of their peripheral blood. And what we saw is that these scribble-deficient mice were resistant to inflammation, and this means that they were more effective at their job when compared to wild-type. But we wanted to know, how does this work? In order to find out a little bit more about scribble, we did RNA sequencing. RNA sequencing is an unbiased bioinformatic approach, which allows us to profile different genes to check for dysregulation. What we saw was that the most dysregulated genes were part of the Notch signaling pathway. So as you can see here in our heat map, all of these genes are associated with the Notch signaling pathway. Here we have our wild type mice, and these genes are downregulated in the wild type. But these same genes are upregulated when we knock out scribble. And the opposite is happening over here, where these genes are upregulated, they're downregulated in scribble deficiency. But what is the Notch signaling pathway? The Notch signaling pathway has many functions, but we looked into its ability to aid in T-cell maturation. So in this image here, we have our early precursor T cells. And as Notch signaling comes into play, it allows these precursor T cells to mature to their DN3A stage. DN stands for double negative, and it's referring to T cells that are double negative for CD4 and CD8 antibodies, which I'll get back to in a minute. But I want us to look at T cells' role in immune defense. I think we've all heard of white blood cells. They help defend our body against different pathogens. And T cells are a kind of white blood cell. Specifically, our lab looked into CD4 positive and CD8 positive T cells, which are able to be identified by their specific antibody markers. And they have very distinct roles in immune defense. So So CD4 positive T cells are known as helper T cells, and they activate other immune cells, whereas CD8 positive T cells are cytotoxic, which means that they kill cancer cells. Putting all this together, we know that Scribble has a role in Notch signaling, and we also know that Notch signaling helps with T cell maturation. So this summer, we asked the question, what does Scribble deficiency do to T cell maturation? In order to answer this question, we did a co-culture of OP9 delta cells and bone marrow cells from wild type and Scribble deficient mice. So OP9 delta is a special line of cells. They're mouse bone marrow stromal cells that are engineered to overexpress the Notch ligand delta. We co-cultured those with our wild type and Scribble deficient bone marrow cells taken from mice, and we immunophenotypically analyzed the output of this cell co-culture using flow cytometry. Flow cytometry is a method of analyzing and sorting cells based on their size, their complexity, and by the fluorescent antibody markers that we stain the cells with. So we stained this output from the co-culture with many different kinds of cells, with many different kinds of antibodies, but we're going to be looking specifically at our T cell population. So here we have our flow cytometry results from wild type and Scribble knockout T cells. I'll walk you through it. Hopefully, you remember earlier, I mentioned our double negative T cells, and that is this population right here. Along this axis is CD4, and this axis is CD8. So these cells here have neither CD4 or CD8 antibodies. Looking over here, we have our double positive cells, which express both CD4 and CD8 antibodies. And over here are our single positives with CD8 and CD4 that express either CD4 or CD8 antibodies. Comparing these two graphs, we can see the major difference between them is the amount of double negative cells in our knockout population when compared to our wild type. We have way more double negative T cells in the Scribble deficient cells compared to wild type. We determined that this is because the T cell maturation is being arrested at the Notch-dependent developmental stage. What this means is that when we take out Scribble, these T cells are unable to mature past their most immature double negative stage. So they can't go on to become double positive, and they can't go on to become immature fully positive, single positive T cells. However, we still need to repeat this experiment in order to check for replicability. And additionally, we also want to look at the mouse thymus. The thymus is a gland located above the lungs, which both humans and mice have, and it is the primary site for T cell maturation. We didn't do this before because working directly in the thymus is more complex than just doing a cold culture, and we wanted to get some preliminary data before we moved into these more complicated experiments. But we hope to do an in vitro culture, and hopefully, if our procedure gets approved, an in vivo mouse model of the thymus, and looking at how Scribble deficiency impacts this T cell maturation. And finally, our significance. Our end goal, of course, is to help patients, and by understanding Scribble's function and role in the body, we hope to be able to help patients who are immunocompromised, who get frequent recurrent viral infections. We also hope to help those with autoimmune diseases, maybe patients who just received bone marrow transplants. However, more research, like our future plans in the thymus, is needed before we can come to these conclusions. And this concludes our presentation. I would like to thank my lab PI, Dr. Gonzalez, my day-to-day mentor, Dr. Singh, as well as the Yes4Cure team and my funding support. And I would like to thank all of you for listening, and if you have any questions.